Good day, everyone. Let's begin with a fundamental shift in how we perceive microbial life. Traditionally, microbes have been depicted as independent, planktonic, free-swimming, cells suspended in fluid environments. This image, while not inaccurate, is only part of the story. The dominant microbial lifestyle in nature, and one with profound consequences for human health and industry, is actually the biofilm. Biofilms are complex, surface-attached communities of microorganisms enclosed within a self-produced matrix of extracellular polymeric substances, or EPS. These aren't just random clusters, they are highly organized consortia that exhibit cooperative behavior, differentiated physiological states, and resilience far beyond that of planktonic cells. Understanding biofilms is essential because they are virtually ubiquitous in soil, oceans, pipes, medical devices, and even within our bodies. Clinically, they contribute to persistent infections and antibiotic resistance. In industry, they're implicated in both useful processes like wastewater treatment and problematic ones, such as biocorrosion. As I often tell my students, if you think of microbes as isolated cells, you're missing about 99% of the story. Biofilms are where the real microbial action is. They represent a microbial society, a living architecture with structural, regulatory, and protective features that challenge our conventional approaches to microbiology. Today, we'll uncover how these systems form, function, and resist the very interventions we typically rely on. Now that we've established what a biofilm is, let's explore how one forms. Biofilm development is a multi-step, regulated process, far from spontaneous. The first stage is reversible attachment. At this point, microbial cells transiently associate with a surface via weak interactions like van der Waals forces. This step is exploratory. If the conditions aren't right, the cells detach and move on. When conditions are favorable, cells transition to irreversible attachment. This involves the use of specific adhesins, molecules such as pili and fimbriae that create a strong bond to the surface. This marks the commitment point in biofilm formation. Next, we observe microcolony formation. Attached cells begin to proliferate and start producing EPS, the extracellular matrix that defines biofilms. This matrix, rich in polysaccharides, proteins, and extracellular DNA, not only glues the cells together but also forms a scaffold for building more complex structures. As the biofilm matures, it doesn't just grow in mass, it organizes itself. Channels and voids emerge, allowing nutrients to flow and waste to exit. These aren't random formations, but functionally beneficial architectures shaped by both physical forces and genetic regulation. Finally, the dispersal phase allows cells to exit the biofilm and colonize new environments. This is crucial from both an ecological and clinical perspective. Dispersed cells often retain enhanced traits, including resistance and virulence, making them potent agents of secondary infection. Pseudomonas aeruginosa is a classic model for studying this progression. Its biofilm life cycle is well characterized and offers insight into the regulatory pathways that underpin these transitions. Each phase of biofilm development is a target for potential intervention. Understanding them is essential to managing both beneficial and harmful biofilms. The extracellular polymeric substance, or EPS, is fundamental to biofilm structure and function. Far from being an inert slime, the EPS matrix is a dynamic, heterogeneous mix of polysaccharides, proteins, extracellular DNA, EDNA, and lipids. It acts as both glue and armor. Structurally, EPS holds the biofilm together, enabling it to form stable, three-dimensional communities. It also forms a protective barrier, shielding cells from antibiotics, immune cells, and environmental stressors like desiccation and predation. Functionally, the EPS matrix supports nutrient capture and cycling. Enzymes embedded in the matrix break down environmental compounds, making nutrients more accessible. The matrix also traps EDNA, which plays a dual role. It's structural and a platform for horizontal gene transfer, enhancing genetic diversity. Different bacteria produce different EPS components. For example, Pseudomonas aeruginosa makes alginate, while Staphylococcus aureus produces PNAG. These substances not only differ biochemically, but also affect how biofilms respond to host defenses and antibiotics. In essence, the matrix is more than a scaffold. It's a living interface between the biofilm and its environment. Understanding its composition and roles is key to disrupting biofilms therapeutically and industrially. Biofilm formation is not just a physical rearrangement. It's accompanied by profound physiological changes that distinguish sessile biofilm cells from their planktonic counterparts. One hallmark of biofilm physiology is heterogeneity. Within a mature biofilm, steep gradients of oxygen, nutrients, pH, and waste products form due to limited diffusion and local consumption. This results in diverse microenvironments, where bacteria exhibit distinct metabolic and growth states depending on their spatial position. Cells near the biofilm surface may remain metabolically active, but those deeper within often experience nutrient depletion and hypoxia, leading to slowed metabolism, dormancy, or persistence. These subpopulations are especially tolerant to antibiotics, as many drugs target active growth processes. At the molecular level, biofilm cells undergo extensive transcriptional reprogramming. Genes related to motility, such as those encoding flagella, are often downregulated, while genes promoting EPS production, adhesion, and stress responses are upregulated. This shift supports stability over mobility, enhancing survival. Another adaptation is the emergence of persister cells, a small subset of dormant, highly tolerant cells that can survive antibiotic exposure without being genetically resistant. These cells can repopulate the biofilm once treatment ends, leading to chronicity. Overall, biofilm physiology reflects a strategic trade-off. Cells sacrifice rapid growth for community resilience, metabolic efficiency, and stress tolerance. Understanding these shifts is critical for designing interventions that reach and disrupt all layers of a biofilm, not just its surface. Biofilms are more than passive clusters of cells. They're coordinated communities, and this coordination is mediated through quorum sensing. Quorum sensing allows bacteria to detect their population density and regulate gene expression accordingly. In biofilms, quorum sensing controls attachment, EPS production, virulence, and dispersal. For instance, in Pseudomonas aeruginosa, AHLs and quorum sensing molecules regulate genes involved in matrix synthesis and immune evasion. In Staphylococcus aureus, small peptides or AIPs perform a similar function. Quorum sensing molecules accumulate as cell density increases. Once a threshold is crossed, they trigger synchronized changes in gene expression. This makes quorum sensing essential for biofilm development. Cells wait until there's a quorum to decide collectively how to behave. Disrupting quorum sensing, known as quorum quenching, is a promising strategy to prevent or dismantle biofilms. Since quorum sensing coordinates virulence and resistance, targeting these systems can reduce pathogenicity without killing cells outright, potentially reducing selective pressure for resistance. In sum, quorum sensing transforms bacterial populations from individual actors into collective, responsive systems, enabling biofilms to function as a unified whole. One of the biggest clinical challenges posed by biofilms is their remarkable tolerance to antimicrobials. Unlike planktonic cells, biofilm-resident bacteria can survive drug concentrations up to 1,000 times higher. This tolerance stems from multiple factors. First, the EPS matrix slows or blocks drug diffusion. Antibiotics that do reach the interior face metabolically dormant cells, such as persisters, which are inherently less susceptible to drugs targeting active cellular processes. Second, biofilm cells upregulate efflux pumps and detoxifying enzymes, increasing their resilience. Third, the close proximity of cells facilitates horizontal gene transfer, spreading resistance traits. This multifactorial defense makes chronic infections like those seen in cystic fibrosis, UTIs, or on medical devices extremely difficult to treat. Even if symptoms subside temporarily, the underlying biofilm often remains, ready to reignite infection. Effective treatment requires more than standard antibiotics. We must consider agents that penetrate biofilms, disrupt the matrix, or target dormant cells. Understanding these tolerance mechanisms is essential to advancing therapeutic strategies. Biofilms are central to many chronic infections, contributing to their persistence, recurrence, and resistance to treatment. In cystic fibrosis, Pseudomonas aeruginosa forms dense lung biofilms, resisting antibiotics and the host immune system. On medical devices, catheters, prosthetics, heart valves, Staphylococcus epidermidis, and S. aureus form biofilms that often necessitate device removal. Chronic wounds, including diabetic ulcers, host polymicrobial biofilms that delay healing. In dental plaque, Streptococcus mutans and P. gingivalis form structured communities driving caries and periodontal disease. Recurrent UTIs are also biofilm-associated, especially when E. coli forms intracellular biofilm-like communities. These infections are tough to eradicate due to tolerance to antimicrobials, immune evasion, and reservoir formation. Conventional antibiotics often fail, prompting interest in biofilm-specific therapies, enzymes, combination drugs, and physical removal. Recognizing the biofilm nature of these conditions is key to effective diagnosis, management, and innovation in treatment. Biofilms are not inherently harmful. In environmental and industrial settings, they play dual roles, offering benefits and challenges. Beneficially, biofilms are vital to bioremediation, breaking down pollutants like oil and industrial waste. In wastewater treatment, biofilms process organic material and remove contaminants. They're also key players in nutrient cycling in soil and aquatic systems and are used in biosensors and biofilters in biotechnology. However, biofilms also cause biofouling, clogging pipes and equipment, and biocorrosion, degrading metal structures. They pose contamination risks in food processing and water systems. Thus, managing biofilms is context-dependent. In some cases, we aim to support and engineer them, while in others, we need to disrupt or prevent them. The same biological structures that make biofilms valuable also make them hard to control when undesired. Controlling biofilms requires a multifaceted approach, targeting their structure, physiology, and communication. Prevention is key. Anti-adhesive coatings and modified surfaces can reduce microbial attachment. Once a biofilm forms, disrupting the EPS matrix is critical. Enzymes like DNases and dispersin B can degrade matrix components, improving antibiotic access. Quorum quenching, blocking communication signals, can prevent maturation and virulence. Physical methods like sonication or scrubbing can remove biofilms from surfaces, especially in industrial and clinical settings. Biofilm treatment must also address persister cells, using compounds that either activate or kill dormant bacteria. Novel antimicrobials, biofilm-specific drugs, and combination therapies are showing promise. Ultimately, effective control isn't about just killing cells, it's about disassembling the community and preventing reformation. Looking ahead, the field of biofilm research is entering a technologically transformative era. New tools are allowing us to understand and manipulate these communities with unprecedented precision. Omics approaches, metagenomics, transcriptomics, proteomics, are giving us insights into the species composition, gene expression, and metabolic activity of biofilms in situ. These techniques are especially valuable for studying polymicrobial communities, which dominate real-world biofilms. Advanced imaging, including confocal microscopy and live cell tracking, allows us to visualize biofilm formation and architecture in real time. This has revolutionized our understanding of biofilm dynamics and heterogeneity. CRISPR-Cas systems are being repurposed, not just for gene editing, but for biofilm disruption and even programmable cell behavior. Imagine engineering a strain that auto-disassembles its biofilm under specific conditions. Synthetic biology allows us to design microbes with bespoke biofilm traits, whether that's enhanced bioremediation or self-limiting growth. There's also growing interest in host biofilm interactions, how biofilms evade immune detection, trigger chronic inflammation, and influence host microbiota. Lastly, AI and machine learning are being used to model biofilm formation, predict antimicrobial resistance, and optimize treatment strategies. The future of biofilm research is deeply interdisciplinary. As our tools become more sophisticated, so too does our ability to manage, exploit, and coexist with these microbial architectures. To wrap up, let's revisit the central theme of today's lecture. Biofilms are the dominant microbial lifestyle, not the exception. They are organized, resilient communities that exhibit behaviors far beyond what we associate with unicellular organisms. From environmental sustainability to chronic disease, biofilms shape microbial function in every imaginable context. Their unique structure and physiology confer extreme tolerance to antimicrobials, making them formidable clinical and industrial challenges. But they also offer opportunities. When understood and harnessed properly, biofilms can be powerful allies in biotechnology, environmental management, and even medicine. The key takeaway is this: if we're to effectively treat, manage, or utilize microbial communities, we must stop thinking in terms of individual cells and start thinking in terms of structured, collective systems, in other words, biofilms. I'll leave you with this. From a single cell to a complex city, biofilms exemplify microbial mastery of survival. Thank you, and I look forward to your questions and comments. Please like, share, and subscribe if you like my videos.